Yeah, so if you guys have any questions on the techniques that I go over today, just let me know. You can just stop me anytime. But yeah, like I said, today we'll be going over engineering approaches for microfabrication. So let me share my screen with you guys. All right. So engineering approaches for microfabrication, as the name microfabrication implies, it's techniques that allow us to fabricate materials in a tinier scale, in the micro scale. So we're essentially going smaller and smaller, right? So if we look back into history, um, a lot of the electronic devices that we've been using, like computers, they used to be humongous. But as we miniaturize all of these features while maintaining these, what you call it, like functionalities, we can further use, more efficiently make use of these spaces physically. So as you can see on the left side image, this is like a example of a hard drive. In the 1950s, as you can see, it's humongous, right? Compared with this human being's hand. And now more recently, hard drives are a lot smaller, and that really helps us to make computers smaller, but it also runs equally, if not better capacities. And so I'm from a biomedical engineering background. So today I'm gonna be focusing on how we use these microfabrication techniques for biological applications. But just keep in mind that a lot of these microfabrication techniques are useful and heavily used by electrical engineers and other engineering disciplines as well. But yeah, going back to the biological side, let's look at this size scale here. Uh, Super obvious things like buildings, like the human being, the size scale is in meters, right? We're like pretty humongous beings. If we go further down in the size scale, we go down to like tissue level, like different organs. For example, the liver, it's about 200 millimeters. Um, Further going down though, each of these tissues are made up of different or individual cells. And so as we get down to these smaller and smaller cellular levels, we're looking at more of these microscale features. And when we're trying to study microscale features like the cell, we need engineering tools or approaches that simultaneously have that kind of microscale definition and resolution for us to be able to study that. So I'm going to be introducing some of the microfabrication techniques that enable studies of cells and tissues. So a major or one main focus is called microcontact printing. This is like a general overview slide. We'll get into like the nitty gritty of it soon, but just to give you like a brief overview, when we're studying cells, we want to be able to also control the geometric shape of these cells as well, and how they're sort of attached onto our petri dishes or whatever substrate we're using. So if we step back, the way we studied cells in the past were to allow these cells to attach onto hard surfaces like petri dishes. We'll let them grow randomly, and then you could, either look at their DNA, protein expression to study their biology, or you could throw in different drugs to see how those cells respond to certain therapies. But if you really compare those petri dish approaches to the human body, it's totally different, right? A lot of the times in human tissue, cells are aligned or they're structured in a certain way with a certain motive. There's like a reason why those cells are structured the way they are. So in order to mimic that maintenance of cellular order and structure in architecture, we use microcontact printing. So the way we do that is basically, you can kind of think of this as stamping different things onto the Petri dishes. So we have these things called polydimethylsiloxane or PDMS stamps. PDMS is literally just kind of like a silicone material that's rubbery. So yeah, literally think of it as like a stamp. And these stamps have these miniature features in the microscale, whatever shape and size you design it to be like. And on these miniature features, you can put on different proteins. So proteins are what helps the cells to attach onto the Petri dish. It keeps the cells happy. So then if you invert this stamp onto a substrate, let's say a Petri dish, like from before, you can stamp those ECM proteins, the blue here depicted in this schematic, in certain shapes and sizes. And then if you throw cells onto it, the cells will adhere onto that pattern shape, as you can see here. So this schematic shows what the size feature of the stamp was, and that feature is closely mimicked in how the cells actually adhere, right? And this is very unique because now we can like control the geometric shape of the cells. And although it seems trivial, if you control the shape and size of cancer cells or any kind of cell, it's been shown to regulate the biological mechanism in a different way. So this kind of method uniquely allows us to study that kind of biological phenomena. So in order to make microcontact printing happen, there's multiple techniques, or actually two techniques that are used in chronology. So the first step is called photolithography, and the second is soft lithography. And we'll be going through those in chronological order. But if you look at the schematic, it's basically the first, these four panels A, B, C, D are part of photolithography. And then E and F represent soft lithography. So photolithography, you can think of it as making like this master mold that will enable us to make the PDMS stamps that I talked about in the earlier slide. And soft lithography is making those PDMS stamps from this master mold that you made in photolithography, and then actually applying it onto cell systems or any system. So the procedure for photolithography, the first part, this procedure is carried out in a specific special facility called the clean room. And the clean room is regulated so that the amount of pollutants like dust particles, literally any particles, the number of those particles are controlled in those rooms. So as a general comparison, regular room air has as much as 35 million particles per cubic meter. And that's a lot, right? Clean rooms, it depends on the grade of the clean room. So here I listed two grades of clean rooms, class 100 clean rooms and class 10 clean rooms. Class 100 clean rooms will decrease that number of particles to 3,500. That's a significant reduction, literally meaning that the room is really clean. And then class 10 even further, it'll be only 350 particles per cubic meter. So how the clean room kind of controls and regulates the number of pollutants or particles is through the specific airflow system. So they make use of regulated gas flows within the entire room so that we can control the number of particles. Another major factor is human beings, us, right? On human bodies, there's actually a lot of particles, dust particles on us. So in order to work in a clean room facility, before we enter the room, we have to gown up into these like bunny suits so that we prevent any of the dust particles that are coming from our body to be polluting the clean room area. So whenever we do photolithography, we literally look pretty silly in these like bunny suits. But that kind of safety precaution allows us to maintain the cleanliness of the clean room and ultimately maintains our finished product to be clean and dust-free as well. So in the clean room, how we go about photolithography is first, we have a specific design in mind. So let's say you wanted to make a PDMS stamp with specific features. You need to be able to really pinpoint what those features look like. And after that conception, what we do is we design those features in a computer-aided design program, a CAD program, right? So that's what you learned in the previous sub-cohort meeting, but it's similar to this. There's a lot of CAD softwares out there, but you just draw out and design your specific features that you want. And these designs are then printed onto a polyester transparency, as you can see here. So this process usually is done by specific companies, manufacturers, and it has a high dot per inch resolution so that we can get very clear-cut micro-scale features. And usually the resolution is around tens of microns, if not better. So this is what we call a mask, a photo mask. The other component that we need is the silicon wafers, as you can see in this image. So silicon wafers are literally just thin slices that are made up of semiconductor material, and it's highly pure, crystalline, and brittle. So we really need to be careful when we're handling it. It's really easy to just snap them in half. And it comes in different sizes, ranging from 1 to 12 inch usually. And they're usually circular, as you can see in the photo here as well. And the thickness is also controlled, usually within the range of 200 to 1,000 microns. But the objective now is to, on the surface of these silicon wafers, we want to etch the design that we drew in the photo mask, like these designs, literally onto these silicon wafers so that it can serve as a master mold later on. So in order to do that, the first step is to spin coat a thin layer of a material called a photoresist onto these silicon wafers. So the general procedure is shown here. So let's say this substrate here is the silicon wafer, and it's gonna be mounted onto this chuck, which is this machine here. There's gonna be like a vacuum pressure so that the silicon wafer is literally bound tight onto the chuck. And then we're going to apply the solvent solution. In this case, this solvent solution is called a photoresist, and it's this red solution that you can see in this image. And spin coating is a technique where literally this chuck is going to spin around at high speeds. And what that enables and achieves in the end is that it's going to flatten out the solvent solution that was just deposited on top of the silicon layer. And using centrifugal forces, we're going to literally make like this flat, thin layer of the photoresist covering the entire surface of the silicon wafer. Does that make sense? Okay. Anyway, once we have that layer of photoresist that's covering our silicon wafer, now we're going to expose our photoresist to UV light. So before we do that though, we use this machine called a mask aligner. So this mask aligner allows us to literally align our photoresist covered silicon wafer. And then on top of that, we're gonna have the photo mask, the one that we printed from earlier on with the desired features. Once the mask and the wafer is aligned, then from the top, as you can see here, we're going to expose UV light onto it. So now what happens is the UV exposure is only going to be exposed in areas where the mask has those features or dead space, the negative space, right? All of the black areas in the photo mask is going to block out the UV light. Photoresists, once these UV lights are being exposed to, photoresists have this unique feature of, we call it cross-linking. So what used to be liquid becomes a solid. So upon UV exposure, selected areas of the photoresist that are exposed from the photo mask, so these areas here, at least in this specific schematic example, they're going to cross-link and they're going to harden. So then the end result is going to look like this, right? These dead areas, it used to have a thin layer of photoresist, but UV light didn't touch that area. So the photoresist there were still liquid. So after we submerge it into a developer solution, it's going to wash away. The only areas of the photoresist that are going to remain is these photoresist areas that were exposed to UV light. And so then in the end, after we develop these photoresists, the silicon wafer is going to look something like this. It's going to have any of the design features that you drew in the computer-aided design software initially. And if you look really closely, this is what it looks like. This is a picture taken with a scanning electron microscope, which is a very high magnification microscopy technique. And you can see that you have like these very fine details in the micro and even nano scales. So to the naked eye, it kind of looks like this. It's like these macro features that kind of just looks pretty in like the chrome light. But if you really look in the micro scale, there's definitely features. So now that was photolithography in the clean room. Now that we have these silicon wafers with desired designs etched onto it, we can now move out of the clean room and use it for making PDMS stamps. So this part is called soft lithography. So like I said before, PDMS is a silicone material, right? It's very, when you first make the PDMS solution, it's this very viscous material. It's kind of like very thick honey. PDMS though, it hardens when you put it in an oven that's at high temperature. So making use of that property, first, we're going to make this honey-like PDMS structure, and then we're gonna pour it onto the wafer that we made from photolithography. Then we're going to cure it. So curing PDMS literally means baking it. So sticking the PDMS into an oven at high temperature, usually at least 65 degrees Celsius. And then once the PDMS hardens, it's like a more rubbery silicone material. So then we can peel off the PDMS, as you can see here, peel off the PDMS stamp that's hardened from the silicone wafer. And then we can cut our PDMS stamps into smaller bits so that we can use it as a stamp. So if you look at this schematic, hopefully it makes a little more sense. So master literally means this is the silicon wafer, and these pink features are, when you look at it from the side cross section, right? The pink features are the designs that you etched onto the silicon wafers. You're gonna let PDMS pour over and allow to at least like geometrically fit into these microscale features that you made with photolithography. And after you pull off this PDMS stamp from the silicon wafer, you have this PDMS stamp, right? So now we're gonna use this PDMS stamp to control cellular architecture. And a general overview is shown here. Going from top to bottom, this is a recap of the last part. So the black area is your silicon wafer, that's the master mold. And the blue area is your PDMS stamp. Once you peel off your PDMS stamp, you invert the PDMS stamp so your features are facing upward. And you're gonna drop a protein solution shown in red here onto that feature of your PDMS stamp. So then we're literally allowing these protein solutions to absorb on the surface of the PDMS stamp. And after you dry the stamp, you have this thin layer of protein left on the stamp surface, right? And again, we're gonna invert the stamp back and stamp it onto a cover slip or any sort of substrate that you want to put your cells on. And then the principle of stamping comes here, where after you peel off your stamp from the cover slip, what's left on the cover slip is then selected features, right, of specific shape and size. And now that this cover slip has, these very selective areas of protein adherence, then the cells, once we put our cells on top of this cover slip, they're going to preferentially attach onto these red areas, onto the areas with proteins. And that's how we can control cellular architecture, as you can see here. So this is one application for microcontact printing, but that is to control cell geometry. So on the left, I'm showing these are cells that have not been patterned at all. So let's say I'm gonna randomly throw these cells onto glass cover slips with no patterning at all. Then they're going to adhere however they like. And even though these two are the same cell type, it looks really different, right? Their geometry is totally uncontrolled. In an engineering approach, we do want to control, to a certain degree, we want to control and confine these parameters like cell shape and geometry. So that's why we're going to use microcontact printing. And as you can see here, you literally have the power to decide what kind of features you're going to etch onto your PDMS stamp. And that starts from the first step, right? When we designed those photo masks in the computer-aided design in the CAD software. So you pretty much have the liberty of determining what kind of shape, what kind of size these stamps are gonna have. And that's how you can control the specific shape and size of the cell ultimately, as you can see here. So from the top row, it's just the square, and then you just elongate the square or into a rectangle more and more. And as you change these features of our microfabrication technique, then the cells also adapt in terms of geometrical shape, and they change their biology as well. Another application, not microcontact printing, but from the first part, photolithography, where we etched your design onto silicon wafers. Another application for that would be to make these things called microfluidic devices. So this is a very popular approach in engineering, in biomedical tissue engineering, at least. And we're gonna make use of the fact that we can control shapes in a micro scale with really good accuracy in order to make these microfluidic devices. As the name kind of implies, these devices are micro scale fluid devices that work with fluids. And to do that, we have like a bunch of these small microscale channels. And these channels are where you flow your fluids in. So the fluids could literally just be fluids that have cells, or it could be fluids with drugs dissolved into it. The possibility is endless. And then we have these very complex patterning of the fluidic device, as you can see here, with different colors. And that's how you can achieve in a single device, mixing, for example, multiple different types of cells. So it could be the study of cancer cells and how they interact with circulating blood cells, or how they interact with healthy tissue cells, but also how those cells in the device react to different drug treatments. So these fluidic channels can literally be used as conduits for flowing drug at certain concentrations, because yeah. So, I just want you to appreciate that because we can precisely control these microscale architectures using photolithography, then that allows scientists to be able to do complex things with the living tissue systems as well in the device. Another application, so this photolithography and soft lithography approach, as you might guess, because we have to be working in like a clean room, we're working with these like very sensitive materials, it's very difficult to master. It's also pretty time consuming, right? The whole process of making those silicon wafers takes a while. So it's very labor intensive. And as an alternative to that, that's a little faster. Although we lose some of the resolution, there are other approaches to microfabrication. And one of them is 3D printing. And you're all probably familiar with 3D printing, so I'm not going to go much in depth, but just realize that 3D printing is like a bottom-up approach. So literally, you have this nozzle, right, which acts like a printer. And from the nozzle, it's going to extrude these filaments or whatever material you want to use. And it's going to deposit that material at the bottom. So from the bottom, it's going to just keep building upwards until you achieve your final, device design or whatever you're trying to make with the 3D printer. So this is by controlling the path of these bioprinting nozzles, we can make microscale features as well. So take example, the microfluidic designs as well. We can make these like channels using 3D printing. Currently though, with the techniques that we have, 3D printing is really great because we can make these like macro scale devices or objects. But if you really look closely, there's always like these like rugged edges. The resolution is just not great, right? So another approach, it's still faster and a lot more cost effective than photolithography. But another approach that has a little better resolution is micromilling. And so as opposed to the bottom-up approach that 3D printing has, micromilling is a top-down approach. So it's literally the opposite. So a typical, this is like a micromill that's commercially available and very easy to use. So it's very small. It's actually like, I guess if you compare it to like this desktop screen size, it's not that humongous, right? So it's comparable to how big a 3D printer would be. But the idea is in this micromilling machine, there's this end mill, and this drill bit is going to literally hash out all the negative space in a stock material that we have. So yeah, we start with like this block of, usually in our applications, a block of plastic material. And then using this drill bit, we're going to create a final design by literally etching away at the negative space. And so how this drill bit moves is controlled by the computer. So that's why a lot of these commercial mills are called computer numerical control machines. And that's because you literally design your device that you want or your end product to look like in a CAD software, very similar to 3D printing and photolithography, right? We are going to transfer that information into your milling machine, and that's gonna move the drill bit in a certain way. And then you end up with this device. But micromilling and 3D printing, because it's really fast in terms of making the end product, we call it it's cost effective as well as rapid prototyping method of making devices. So one application, again, if we go back to the microfluidics example, we can also make microfluidics using our milling approach. So these are actually microfluidics that were made not from photolithography, but milling approaches. And sure, the resolution or the size scale is a lot bigger. So these microfluidic devices are a little bigger than what you would expect to make from a photolithography approach. But the functionality is there. And that's what this panel is trying to get at. So this is these two areas is the actual mold that was milled out from a stock material. So you see these like small features. I hope you can see like these like channel-like features, right? Those are actually protruding out. And then once you pour PDMS onto these molds, so this is the soft lithography technique, right, that I went over earlier. We can very similarly make these PDMS casts, which is shown above here. And the features, the channel features, are exactly replicated in these PDMS devices as well. So it resembles what we have in these master molds, right? Once we use these PDMS replicas, so this is one example, right? On the left column, we have a device that has these very complex miniature features on the top of the channel. And what that does is it's gonna make turbulent flow when you flow liquids through it. So let's say we have, as shown in this photo, we were flowing food dye. That one channel is gonna be yellow, the other is gonna be blue. Once we flow them in the beginning near the inlet where the channels start, you can see a clear separation of the colors. But because of these miniature microfabricated structures in the microfluidics, throughout the channel, the two colors are gonna mix, and the end result is gonna be this, where it's just all mixed and it's all green. As opposed to a control device where you don't have these miniature features, you literally just have this channel. So there's the theory is that there's not going to be any turbulent flow, so there shouldn't be any mixing, right? Same thing, if you flow the yellow and blue food dye, it's going to remain separated throughout the entire channel. So this kind of application goes to show that, you know, milled microfluidic devices are still very functional, and it achieves the purpose that it's supposed to, right? And that's because we are able to precisely control these microscale geometries in the devices that we make. Yeah, so those are some of the microfabrication techniques that I kind of wanted to introduce to you guys. There are a lot more in the field, but I think at least the photolithography and soft lithography is probably the most widely used and most popular approach. Now, what a lot of labs are doing is we're trying to look into faster ways, more cheaper ways, while maintaining the integrity of our science. So a lot of us are exploring these milling and 3D printing approaches. So I wanted to introduce those to you guys. Yeah, that's it for what I have in terms of microfabrication techniques. Do you guys have any questions? I know I went through a lot really fast, but if not, I think we're good then.
Thank you, Ta.
No problem.
All right. Bye, everyone.